Yeah, welcome everybody. I'd love to introduce Jianhan Chen from UMass Amherst, and he's been working on simulating underlying the dynamics of cells and the chemistry within them. Yeah, welcome, Jianhan.
Okay, thank you, Greg. So I'll get started. So thank you all for joining, and I think this talk is going to be quite different from the many of the talks that you have listened to on the series. So I will try to take it slow, and please interrupt me anytime you want, and it'll be great to have a good discussion. So today, what I'm going to talk about is some of the strategies that people have used to simulate or study the phase separation phenomena that occurs in cell. So first, just a little introduction to phase separation. My so-called liquid-liquid phase separation actually is a really well-known phenomenon in physics. So these are thermodynamically driven and reversible mixing and demixing of solution into two distinct liquid phase, where you have really different solute concentration. So this can happen whenever the balance between the solute-solute and the solute-solvent interaction is such that they are sufficient to overcome the entropic cost of coming together of demixing. So in this little movie that was playing earlier, so these different colors are different peptide chains, and so we start out from a homogeneous solution, because the peptide interaction are strong enough that they will come together and form this little droplet in the middle. So this little droplet is a more concentrated liquid droplet of the protein solution. And this happens because the protein-protein interaction is strong enough to overcome the entropic cost of coming together. So this type of phenomenon is really well known, and there are this little equation shown here is called Freud-Hagen theory for describing this kind of phenomenon. And this is really a really well-known phenomenon, and it can happen most frequently observed with polymers and other solids. So to really think, this is really kind of a really fundamental phenomenon in physics. Oftentimes, to understand this kind of phase separation, we would draw a so-called phase diagram, where you show the existence of different phases as a function of the solute concentration, which is the x-axis. Then on the y-axis is some type of control. It could be temperature, pH, or anything that could perturb the phase equilibrium. So what really happened here is imagine the y-axis is just temperature. So and if we pick, say, the temperature represented by this line, let's say this is room temperature. So at really, really low concentration, like a state 1, right? The concentration is really low. You get a single homogeneous solution, right? Which is kind of illustrated down here. So as you increase the solid concentration, the solid could be polymer or proteins. And once you cross this phase boundary and enter into the middle, so this actually is a region where a homogeneous solution will become unstable. So they will spontaneously separate into two regions, right? Really, one point is here. So really only on the phase diagram, only these 2 points are stable. So if at this concentration, you will phase separate into a large portion where you have a lower concentration. Then the other phase, you have a higher concentration. So this is CD. So what you experimentally can observe is that these droplets start to show up in your solution. Now, as you further increase the solid concentration, and you're just going to, the two phases still persist, what can happen is that you just introduce more and more a dense phase. And eventually you cross over to the other phase boundary and enter into a really high concentration. Now you re-enter this one phase region where you only observe a single homogeneous but dense phase. So this kind of phenomenon is really well known. It's somewhat kind of, you can say, it's boring in terms of biology, but this is also well known that even protein solutions can undergo phase separation. This phenomenon have been observed since like 1949, and so the papers I kind of include here are some studies from 70s. So Ishimoto and Tanaka, they have shown that the lysozyme protein solution, as you increase the salt and the protein become more sticky, the repulsion between proteins become reduced. They tend to stick to each other, and they will eventually demix in the phase separate. So this is really kind of known that the proteins can do that. And later on, George Benedek from MIT, he actually studied quite extensively on how this alpha crystalline, this is a protein in our lens, can phase separate, and he studied this phenomenon, showed that this is really follow the classical liquid phase transition. So far, and this in terms of phase separation of protein solution is really more limited to polymer physicists or physicists. So most people, yes.
So yeah, kind of bring this down to kind of the core intuitions. I mean, this is like precipitation of like if something like salt dissolved in water, and now this is salt falling out of solution. I guess that's a liquid solid in that case.
No, here actually does not. This is a liquid phase separation, so it's not liquid solid phase separation. So what happened here is that as you increase the salt concentration, or at the sufficient high salt concentration, the sufficient high protein concentration, and the protein will come together and form a more condensed but still liquid phase. It's like what you see under microscope is that you form this droplet suspend in a more dilute phase or more dilute solution.
Okay, so this follows like you're forming bubbles of one solution inside the other solution.
Kind of, yeah. It's except that the bubble is a really condensed phase. You can think about like in ocean, right? You can have this flow of really high concentration, much more concentrated salt water. Right? They can. What do you call those things?
Right, like Gulf Stream is a saltier flow.
Yeah. So this is still a liquid, right? There's no solid or aggregation formation, and this process is fully reversible. So as you increase the, for example, if I go back to this phase separation, right? So imagine if you are here, right, at the concentration 2, and it was phase separate into this droplet, and this is more condensed, more dense phase, and this light green, the more dilute phase. And as if you increase the temperature, right, eventually you will enter the single phase regime, or this droplet will dissolve. So you can crank up and down. This process is fully reversible.
So then what causes it? Like if I'm imagining like water, it's okay, there's the hydrophobic, hydrophilic things. So I can imagine how you might mess around with water, but what causes something with high concentration to clump into with things of its own like type?
Right, that's a great question. So this is really in the simplest ways to understand this process is using this particular equation. This is called Floyd-Hagen theory. So this F is a free energy of a phase, right? A particular phase. So the first two terms, you can think about this phi is a fraction. You can think about this as a concentration. So essentially, you have entropy of the one phase, entropy of the other phase. Then this how do you call it? A sigma, right? This sigma is the interaction between the solute and the solvent. So if this term is strong enough, so if the solute and solute interaction are strong enough, so when they come together in the form of condensed phase, so what they need to overcome is the demixing entropy. So if the protein can stick to each other stronger enough to overcome the entropy of forming a more condensed phase, then this would happen.
So entropy is a tricky one. I guess it's I'm not visualizing what causes this, or what's the causal driver of entropy. Like if we're thinking molecular dynamics, what makes molecules want to be mixed versus not want to be mixed?
So molecules always want to stick to one another, right? So it depends on how strongly they stick. Let's say, imagine that let's say your favorite protein at a particular concentration, they want to stick to each other with 1 kcal per mole affinity. Right? Of course, if they don't stick to each other, they will never phase separate. But let's say they stick to each other with 1 kcal per mole. But when they stick to each other, they also cause entropy, because the entropy want every protein to to flow around, right? To occupy, to sample as much of the volume. So at a particular concentration, so the entropy cost, the delta S, right? We can at the zero order approximation, we can think about as a fixed number. Then the affinity of the protein sticking to each other is also a fixed number, delta H. So then we can then whether delta H or delta S wins is determined by the temperature, because the entropy contribution at T delta S in the front, right? So temperature is it determines, it's a dial that you can dial that controls the importance of the entropy. So this is really what happened in the simplest, in really zero order approximation. This is what happened here. So if you are sitting at this particular concentration, and as you increase temperature, then the demixing entropy become more and more important. They will eventually become a single phase. They cannot phase separate. But as you decrease the temperature, the importance of the entropy, right, it will become smaller and smaller compared to delta H. Then at a certain point, the delta H or the protein-protein interaction or the polymer-polymer interaction enthalpy, it's enough to overcome the demixing entropy. Then they will phase separate.
Okay, so the way it seems like the underlying dynamics work like this: you have a bunch of proteins that may want to stick to their own variety or the other thing that they're mixed into, and the higher the temperature is, the more it bounces them around randomly. That's the entropy.
Yeah. That's
also,
one way to think.
The higher the concentration, so the lower the concentration, the more paths there are to meander around. If it's a high concentration, all the paths are blocked, so the impact of higher temperature is blunted.
Right. So I would think about the path. I would think about this is the average mean free path, right? So essentially, it's the effective free volume of each protein. It becomes smaller and smaller as you go to higher concentration.
Got it. So it's a lot like, tell me if this analogy is also right. Like a nuclear reactor, you have neutrons that are flying around because of reactions, and their mean free path until they hit another fissionable thing. And if you pack it densely with a bunch of like control rods, the mean free paths are blocked.
It's like in a random diffusion, mean free path is determined by the concentration itself.
Got it. Yeah, that makes a lot of sense. Thank you.
Yeah, super. That's a really good question. So these phenomena are really fundamental, right? As you can imagine, any molecule can stick to one another. They have an opportunity to undergo phase separation at the appropriate condition. But people do not feel that this is terribly relevant to biology, other than in the eye crystalline, because people know that in early formation of development of our eyes, right, this process is really important. Our eye lens only grow once. So this actually was a lot of interest in really limited scope. But one of the major discovery actually was made in 2009 by Clifford and Hyman. So what people know that there's something called there are a lot of so-called protein RNA bodies in our cell. So such as the stretch granule or nucleoli that happened when the cell under division. So people know that there are these bodies, right? These clumps of cellular bodies that mostly consist of proteins and RNAs, and in most cases, like in our cell organelles, they will have a membrane surrounding our organelles. But these so-called cellular bodies are just a different class. People know that they exist. They are a pretty important function, but they do not know what they are. So the major discovery was made in 2009, is that what they show that these, for example, these P granules, what they show that these bodies are actually liquid-like droplets in our cell. For example, they show that they can merge, they can wet the surface, or they can. The more important, they show that they appear to form, condense, or dissolve as a function of the concentration. So this really is similar, or this really resemble the liquid-liquid transition. People know that it's a really fundamental process. And they later on, they show that this nucleoli, which is another type of protein RNA bodies or collection clumps, also appear to have liquid-like behaviors, right? So what they're trying to show here, they can kind of, you see these two droplets, they come and merge together, like, or you up here. So as a function, they can merge, or they can split, they can dissolve and condense. So this was really a major discovery. And later on, like Mike Rosen showed that this phenomenon actually is even more general than not just those protein RNA bodies can do this. And he showed that this is so-called protein-rich motif, and this is S3 domain. So these are some signaling molecules in cell that do something important. But what they show is that because each of these motifs can bind to each of this red block can bind to this yellow thing. And what they show is that if you put multiple copies of these binding motifs, so such that you can introduce so-called multivalency, which means that each molecule can form more than one interaction. And if you do that, he showed that they can, when you put these two proteins together, they can also undergo a liquid-liquid phase separation. So essentially, engineering this ability for molecules to interact with one another, they will have the opportunity to overcome entropy and demixing entropy and the phase separate. And he also showed that these are indeed liquid-like droplets. They can merge, they can dissolve and condense reversibly. So since then, there have been really a lot of excitement in the field in the last 10 years. People now discover that this is really really prevalent phenomenon in biology, appear to play important roles in many different biological processes, and have been implicated in different kind of human disease and disorders. So there have been really strong interest in the biology field to study this phenomenon. So, and at the same time, people, what we really recognize is that oftentimes these also, so they actually make up a new term called biological condensates, as referred to all these droplets or droplet-like bodies that are formed through phase separation that play some certain roles in biology. So what people realize is that the major drivers of the formation of these biological condensates are dynamic and the multivalent biomolecules. Right? Like this example, right? This will be when you put the multiple interaction domains on a string, they are multivalent, they are dynamic. And whenever you have this kind of molecules, which is really are polymer-like, they will have the tendency, or under certain conditions, they will phase separate. And people start to look at how different type of physical interactions are really key, right, to bring this molecule together when they phase separate. And so these are the details we do not need to talk about now. But the key thing is that the major drivers of these biological condensates are dynamic and multivalent biomolecules. And there are two major classes of dynamic and multivalent biomolecules in our body. One class is known as intrinsically disordered proteins. I'll get to this a little more. The other one is RNAs, right? RNAs could be messenger RNA. Often our body has a lot of RNAs, right? The COVID vaccine, it's an mRNA. And so in order to understand the mechanism and the processes that are involved in the formation of biological condensates, and trying to understand how different cellular factor or even a drug molecule can control the formation or the properties of these kind of condensates, like theory and the simulation have really playing a really important roles. As you can imagine, a lot of early theories that are used to describe polymer phase separation are applicable in certain ways to understand biological condensates. But then in order to understand all these detailed factors, imagine that if in one of the key drivers of condensate, if you have a certain mutation or disease-associated mutation, and how is that going to affect the phase separation behavior, or affect the condensate property, or when you change a cellular condition like increasing pH or increasing salt, or when you introduce some drug molecules, and how can them interaction of the presence of that molecule can control or modulate the phase separate property. And these kind of detailed questions are really cannot really be handled by theory alone. So theory will tell us some general principles, how things could happen. But if you really want to predict a specific impacts of mutation, drug molecule, or certain cellular signal, really simulation, it's, I would say it's really required and to help understand various experimental observations. So
So to understand the
yes.
dynamics, like how they're different in this regime, is it fair to say that the interactions become a lot more quantized? I mean, I'm visualizing. Normally, we think of a bunch of proteins of multiple species that are floating around
Uh-huh.
free. Proteins hit each other by accident, so you have a certain reaction rate, or whole droplets of the different materials are colliding with each other, creating a much higher concentration transiently. It creates a very different reaction rate.
Absolutely. So the properties of the condensed phase actually is a topic that is being under really intensive investigation, right? So people initially thought, like, because in early work, right, in Hyman and Lawson's early work, we see that these cellular bodies are liquid-like. But now we actually now it's pretty clear that these are not classical Newtonian liquids. So these are really viscous liquids that have really complex material properties. For example, they are often viscoelastic. Many of these condensed phase actually eventually undergo so-called aging process. They will either form a gel, or they can aggregate, or they can lead to amyloid fibril formation. So there is a lot of really interest in understanding what exactly the condensate look like, and what's the impact on the structural dynamics and the interaction of molecules within the condensate.
All right, that makes sense. Thank you.
Okay, so before I try to introduce how the field and also us have been trying to use molecular dynamic simulation or use modeling simulations to understand these processes, I kind of want to just introduce this class of proteins. I'm not sure everyone is well aware of these proteins. These are so-called intrinsically disordered proteins. So obviously, AlphaFold, right, from Google has made huge impact. I think it's really one of the most important breakthrough that happened to the field in the last few years. So now we really have the capability to predict the structural properties of almost any folded proteins we will probably ever encounter, and the accuracy is it's really amazing. But on the other hand, there are a lot of proteins in our proteome or in our body. They do not have well-defined structures, right? So these class proteins actually was also discovered somewhat recently. I think I would say just mostly this century. In the last 2 decades or so, it's really becoming well recognized that these proteins are very important. They are really prevalent. So the signature of this, the key property of this protein is that they do not have a stable tertiary structure that the tools like AlphaFold and others are trying to predict. So these proteins are referred to as intrinsically disordered. It's really trying to highlight the point is that it's the sequence signature of this protein that determine the conformation to be dynamic and heterogeneous, and not because we are expressing these proteins in a non-physiological condition. So, and the people have actually now, this is pretty well understood. So these proteins often are enriched with charge or polar residues. They often do not have a lot of hydrophobic residues that are required to drive the hydrophobic collapse for the protein to fold. And the important thing I really want to highlight is that this class of protein represent about 1/3 of our proteome. And more importantly, these proteins play critical roles in certain cellular function, particularly cellular signaling and regulation. So for example, about 2/3 of the proteins that have been annotated to be cancer associated have been classified as intrinsic disorder protein. So these proteins, these molecules really are the key switch or communicator of our cellular network. So there are different ways, right? We can try to understand why nature choose to use this type of messy protein, right, and to perform such important function. So that's probably for some other talk. But the key thing I want to highlight is that if you think about the protein sequence structure function paradigm, right? So what we say is that sequence determine the structure provide the basis for function. So this is why breakthroughs like AlphaFold2 is really transformative. But then for intrinsic disorder proteins, now the meaning of the structure really need to be dramatically expanded, right? Instead of thinking about one or a few structures to represent the conformational state of the protein, now we need to think about a dynamic and the most really likely heterogeneous ensemble of structures. And to really, we need such an ensemble to describe the conformational state of this disorder protein. And then how these proteins perform their function is really encoded in the manner of these ensembles will respond to different cellular signals, such as binding of a cofactor or binding a drug, and how they respond, how the ensemble responds to these different perturbations, really encode the molecular basis of how IDP function. And it has been now established that these disorder proteins can do all kind of fancy things. For example, they can actually fold into a more stable structure upon binding appropriate target, and they can fold into different structures depending on what they encounter into. Or oftentimes they actually can remain highly dynamic when they are bound to a specific target, right? They form so-called dynamic complex. Or there's another word that the people in the field also use called fuzzy complex. And so for the purpose of this talk, we are mostly interested in the ability to self-assemble of really phase separate, right? And as I briefly mentioned early on, many of the IDPs, after they phase separate, they can undergo aging and transitions, right, from a gel, or they can aggregate, etc. So these are really really interesting protein, and play really important role in phase separation. And as you can kind of imagine, these are also really complex proteins to describe in computation. So as I mentioned, molecular simulation and the modeling simulation have been really, I would say it's really critical if we want to understand all these nuance and the details and the specific details of phase separation and how phase separation mediate function. So the key approach that people have been using in the field, I would say these are called physics-based modeling and simulation. So for those who have not heard about this type of simulation, I just kind of want to give a really brief introduction. So there are two key components when we go about simulating biomolecules or any chemical systems. So one of the components usually is referred to as force field, which is really the energy function. It's a mathematical equation that, given the conformation or the coordinates of all the particles in the system, then we want to estimate the energy, right, and calculate the interactions of the system. So that's the force field. And then the other component of in molecular modeling and simulation is so-called referred to as sampling, right? So we need to there's a calculation of the dynamic trajectory. So we need to be able to sample all different ways that this chemical system or molecules can organize themselves. So these are really two key components. So if you think about the model, if you're thinking about the models that people have used, so people have designed really different level of models, right? So at different resolution. So in early days, people use this so-called lattice model. So all this called HP model. This actually has been really also, it's really simplistic. You just think about, imagine that you have protein sequences. You only classify your residues into either polar or hydrophobic. And this so-called HP model in early days actually teach us a lot about how natural proteins fold. And this actually leads to the final energy landscape theory, and which I think is the what we believe is the mechanism how protein folds. And the more typical model will be the so-called all-atom model. All-atom model we will represent each atom in a molecules explicitly. Of course, if you want to look at enzyme reaction, you can use even higher resolution. You can start to describe valency electrons, so that you can study the chemical reactions. Then the type of energy function, right, we often use to describe this large molecular system like proteins and the nucleic acids. we usually use some type of classical or empirical energy function. We do not try to use quantum mechanics. And this classical energy function is often, as I mentioned, is referred to as force field. There are several popular flavors that are being widely used in the field, like Chen, Amber, OPLS, etc. So this energy function contains terms that are inspired or that are basis that is based on our physical understanding of the type of molecular interaction that exists. For example, we know that there are chemical bonds, and we know that the two bonds, adjacent bonds, the angle of these two adjacent bonds is also limited by the molecular orbital property, etc. So, and we know that for any atoms, right, they can have this so-called van der Waals interaction, or if they carry charge, they can have Coulombic interaction. So this is also why we often refer to this kind of approach as physics-based, because the energy function is an approximation, but the approximation is based on our understanding of the type of physics involved in the interaction of this system. So then, for the generation of for the given how we can describe the energy of the state of the system, then the way that we can use to simulate or mimic the dynamic fluctuation of the system, there are usually two major approaches. One is called molecular dynamics. Essentially, we just solve Newton's equation of motion. So given the force, we can calculate acceleration. Based on acceleration, we can predict how the velocity is going to change. Based on the velocity, we can predict how the position of each atom is going to change. So it's a really, really standard approach. Or we can use something called Monte Carlo. Monte Carlo, essentially, you use a move set to generate a new conformation. Then you use your energy to determine whether you want to accept or reject this move. So for simulation of condensed system, which is really most biomolecular simulations are, molecular dynamics is really much more efficient for different reasons I will not probably get into. So then this type of physics-based simulation is really simple in terms of the fundamentals, but it can be really powerful because it's a really general approach. So oftentimes this is the type of how information is generated from molecular dynamics simulation. I often use this really simple flow in my class when I teach molecular dynamics. So you start off on this energy function, right? X is the coordinates of all the particles you have in your system. Given the energy function, then you can do molecular dynamics and generate a trajectory of your conformation as a function of time. Now imagine if you run this simulation long enough, you will sample all the relevant conformational states. Then you can derive probability distributions of your conformational states. And based on the probability, then you can calculate the free energy, right? Because free energy essentially is just probability in log scale in the RT. So once you have this probability distribution, then you can go back and calculate all relevant thermodynamic properties, such as energy, entropy, or binding affinity, or anything that you can think of, right? So this is a really general approach, and I think the development of this approach has really a lot of impact in many fields, and so the example that I show here is a simulation of a whole bacterial cytoplasm, and this actually is just a crop out region of this of the whole simulation. What I really want to illustrate is that this is a general approach that can be used to look at really complex systems. But the critical limitation is that these simulations can be really expensive, because especially if you represent all atoms in our system, the bond vibration is very fast. This limit the kind of numerical time step when we use to propagate to update the conformation to be about 1 or 2 femtoseconds. So this 10 to the minus 15. But many phenomena we want to study occurred on biological time scales of much greater than microseconds. So this means that in order to simulate this system, we need to run trillions or billions or trillions of steps. Then at every step, we need to evaluate, we need to calculate the force on each of the particles in our system, right? You need to evaluate this energy function. Then the system biological systems often contain many atoms. The bacterial cytoplasm has that particular simulation actually have 100 million atoms. Most of the system we study routinely is not that big, but it's still going to be only order a million atoms or even higher. So this calculation become really expensive really quickly. So the point is that even though this is really powerful, there are critical computational bottleneck that need to be resolved. And I will circle back to this a little bit. People actually trying to use machine learning type of approach to really overcome the computational cost of using molecular dynamic simulation to generate confirmation. So I just want to mention that the D. Shaw's research they actually designed this special purpose supercomputer called Anton, and this actually contains application specific circuits that are designed for MD. And this supercomputer can actually generate microseconds of trajectory within a day. So it's something has been really powerful in the field. So circling back to the study of phase separation. So as you can imagine, so in order to study phase separation, we need to look at hundreds, if not thousands of copies of proteins and how they bounce around, come together and form a condensed phase. So this is really expensive process. And the most commonly used models are people where we need to reduce the resolution of the system such that we can afford to simulate large enough system for a long enough time. And the most popular model is so-called C-alpha only model. So for those who heard about this model the first time, you'll find it to be really strange. So what the model does is that we're going to represent every residue in our protein or peptide chain in our protein to be a single bit, single ball. Right, so we really reduce a lot of resolution, and then these beads, the C-alpha beads, they will interact with one another through some effective potential. So people have used, for example, you can look at the PDB trying to figure out how often alanine come together with another alanine, how alanine come together with another leucine. So we can use those statistical potential to describe the effective stickiness between different amino acid type. And then this becomes the energy function used in this C-alpha model. So it's really simple, but really powerful. So even though this kind of model actually been shown to be quite useful in study of protein folding. And one of the early models actually was developed by Kim and Hammer at the time they were at NIH. And they showed that this really simple C-alpha model with statistical potential can actually do a pretty good job in describing dynamic interaction of proteins. And they can reproduce experimental data reasonably well. So then the first adaptation of this kind of model to study phase separation was done by Jianhan et al. And Robert Bass and the co-workers. And this actually was done in around 2018. And since that, they showed that this model can be used to really describe, capture the sequence dependence of whether a peptide will phase separate or not. And then this became really popular because it's really efficient. Now there are many different variants of this type of C-alpha model for phase separation. And then the main difference is really on how one would go about describing the pairwise effective interactions and how you go about optimizing them, right? So people actually have used some of the more advanced machine learning model, a machine learning approach, trying to optimize these potentials. So here I just, yes.
This looks a lot like molecular dynamics, just coarser. Is that
yes,
a connection?
It is. That's exactly what it is. It's just molecular dynamics using a really coarse-grained representation of the system.
So then what's a ball here? Like at what granularity do you choose your balls?
So each of the ball is one residue, the whole residue, right? If your protein has 100 residue, so it's just going to represent by 100 balls on a string.
So wait, well, what's a residue?
Residue is proteins are polymers of amino acids.
Mm-hmm.
So when amino acid is incorporated into a protein, it's called a residue because when the amino acids get connected to each other, they undergo so-called condensation reaction and they lose water. So whatever is left of amino acid is called a residue. So residue essentially is the monomer unit of protein.
No, yeah, and I guess how long is one of those things, or how do you differentiate boundaries between residues?
Oh, they don't distinguish boundaries. So essentially, they just take each C alpha, right? It's what it's called C alpha model. They just take each C alpha and using that C alpha to represent that whole residue.
No, well, yeah, right. I mean, because the sequence, it looks like it's just one giant protein at a high level, but clearly you
Yes.
chop it up into functional units, I guess. But I'm not sure how you establish those boundaries. Is it like a weak connection where it gets floppy, or some other?
Um. So imagine that you trying to you take a protein, right? Like all this atomic representation. You just start deleting things. You just delete all the side chains. I say I don't care about side chains, right? Then you're going to be left with the peptide backbone chain. Then you say I don't care about the peptide chain either. I'm just going to delete all hydrogen. Then you are left with just CR. Okay. Then you try to remember, say, okay, this CF actually come from RNA. I'm going to assign a certain parameters to somehow still represent some of the properties of RNA. So there's no boundary here. What I'm trying to say is that literally it's just coarse-grained. There's no boundary.
Okay, so I think what I'm confused about
Yeah.
is, to me, that whole thing, the whole sequence of balls looks like just one protein.
Yeah, this is one peptide chain, yes. One
Then
protein.
how do you chop it up into multiple balls?
You mean if you want to model multiple proteins?
Well, here it looks like one protein. It
Yeah,
looks
this one
like a
yeah.
sequence of one protein is broken up into multiple balls. So it seems
Yes.
like another problem to find those separations that are useful. Like how do
Oh, how do
you
you
the position
coarse-grain?
of a protein chain?
So are you asking about how do we coarse-grain? How do we choose
How do you coarse grain? Yes.
That's a really good question, and maybe I'll circle back to it. This
I
actually
see.
is a really essential question on how we design the cost, or what cost grading will retain the most information, or retain the most level. This actually is a really important problem. So here, the CF only model is just a really convenient choice, because every residue has a CF, right? So if you just start to ignore the side chains and you just take the CF, it's a really convenient choice. Now, what I should point out is that it depends on the level of complexity. Some of the CFR model have a uniform size, right? Which is kind of crazy that you're going to make like glycine, which is really small, to be the same size as say tryptophan, which is really large. But but some people you can argue that maybe at least resolution you don't care whether at least the difference is beyond the resolution. But there are also people who actually fine tune the size of the bead, right, to correspond to the size of the amino acid. So these are all become design choice, and most of the time, so this is really empirical, essentially based on your physical intuition, whatever you decide that make the sense. And you can argue that maybe this is not the best way to do it, right? So people actually are working on this more subjective, unbiased way to think about how we can rigorously coarse-grain, reduce the resolution with minimal loss in the information. Usually, when we talk about minimal loss information, we're talking about the partition function or the thermodynamic distribution. Okay.
It makes sense. I mean, this caught my attention because this sounds like it's the main problem of science, what you're describing right now.
Oh yeah, this is
Yeah.
yeah, and this is really, I think it had really great intuition. So there are people actually trying to use really more unbiased way to design and derive this type of representation. And but so far, as I will explain, that our approach is really intuition and kind of based on the guide by physics, and this kind of approach actually also work reasonably well. The reason is because there's so much how can I put this so much underdetermination. So oftentimes we have to bring in physics or physical intuition to make sure that whatever we choose still makes sense. All what I mean is that there is not enough constraint from the data alone to tell you what's the best.
Right. Makes sense.
Yeah. So I just want to highlight, like, this model sounds, I mean, for people who study proteins, and especially people, if anyone from AlphaFold group here is they say this is ridiculous, right? How can you learn anything from this kind of coarse-graining? You're losing all the key information. So I just want to kind of take one of the model called Cavados, right? This is one of, I would say, the better model and it's developed by one of my collaborators, Christian Lindorf, and just to illustrate how people go about optimizing this kind of model. So in this, this is also this Cavados is also a C alpha only model. So Greg, how much time do we should end up at 1, right?
Yeah, optimally. But if you take a few more minutes, it's fine too.
Okay, yeah. So we're about 1/4 through the talk, but it's okay. I think
I can say there's a lot of content, obviously.
yeah. My main purpose, I really want to introduce to this group this problem about phase separation and some of the essence approach you're trying to bring. This is really my main goal. So let's just keep going. So in this particular model, every bead is the same size, and the beads will interact through 2 types of interaction. One is called Lerner-Jones. This essentially is a really common energy function people use to describe the van der Waals or nonpolar interaction between atoms or beads. Then the other is the Debye-Huckel. This is also the most commonly used way to describe electrostatic interactions in solvent, right? So there is this exponential term which is really capture the salt screening of the electrostatic interaction. Then the first portion is really just Coulombic interaction with the effect direct constant, right? So this is really standard way. So the main thing that in this Cavados model is that they use a more systematic optimization approach, right? So it's using Bayesian-based approach. So in this loss function, they have essentially these are the chi-square of certain key parameters. RG is radial gyration, which capture how big these disorder chains are. Then this PRE, which is it's a little harder to explain, but this is some NMR experimental measurements that can capture some pairwise interaction, pairwise distance within the polymer chain. Okay, so essentially you have these 2 chi-square, and this essentially is regularized by some prior distribution of the lambda. Lambda is actually the scaling of the interaction between different type of. Okay, so in this optimization, what you're mainly trying to optimize is the lambda. So essentially, you have 20 lambda parameters, which determines the stickiness of the 20 amino acid, 20 beads that correspond to each of the residue type. So they can use this kind of approach to kind of more rigorously search for optimal parameter. What they show is that this M1 is like they do multiple runs of optimization. M1 is essentially just first generation. They shows how the parameter fall into space and how they can improve the ability to reproduce experimental data. So on y-axis is experimental radial gyration or size of the protein, and on y-axis is the simulation. They show that they can really improve to reproduce experimental size of these molecules pretty well. And once you do that, they show that, so in this other panel, what they show is something called CSAT. Essentially is that during the phase separation, the stability of the condensed droplets is measured by the concentration in the dilute phase. So if you have a condensate that is more stable, the dilute phase concentration will be really low, right? Because more molecules will be sequestered into the condensed phase, and you will be left with a more dilute phase. So the CSAT is the measure of the stability of the condensate. So they show that if you do a good job optimizing this monometric, the single proteins dimension, you actually also end up doing a really good, pretty decent job in predicting how stable your condensates are. So this is a really decent model, really very fast. These models are extremely efficient. You can run it, like it's really cost essentially nothing. And this actually allow Christian to look at the conformational properties of all the disorder proteins in our human proteome, right? So the work actually was now recently published in Nature. And so I'm not going to get into details, but the point is that the design model it's really, really simple, but can be really powerful because it's really efficient, allow you to really look at the whole picture. So in this particular paper, they did some really in-depth analysis of how different disorder proteins involved in different function or localized to different region of the cell might have different properties, and they have some really fascinating insights they learned from this study. So another thing that the example I want to tell you guys a little bit here is that to illustrate how this kind of really simple model can be useful is that there is this Alex Hohaus at WashU. He actually take another CFR model and use that as an input, because as I mentioned, this model is really efficient. You can really calculate even any sequence. You can calculate ensemble properties of this disorder protein really quickly. You can figure out how big they are, what's the end-to-end distance. You can calculate whatever property, whatever conformational ensemble property you might be interested in. Then this can be then used input into this recurrent neural network model to make a predictor. And then you can it just become even more efficient. Right now, you can really go take this predictor called albatross, right? You can use them now to do design, for example. So yet another example. This example I think is probably to me is the most exciting one, and it's done by one of my friend Michael Fike at MSU. So he actually developed this model called a generative model called IDP SAM. And this is actually it's a transferable generative model for generating IDP ensembles without going through the simulation. So the input, the training data actually was not generated by CFR model. The early version of this model was generated by a CFR model, but it's not as interesting because those CFR model is so efficient. You can just run the original model. You do not need a machine learning model to help you speed it further up. So in this particular case, they actually they use an atomic model to generate many ensembles. Then they use these ensembles as input to train a generative model that allow us now to potentially replace the MD simulation. So this is actually something extremely exciting. So as I mentioned earlier, that the physics-based simulation is really a really general and really powerful technique that really could promise, has the potential for us to figure out the disorder ensemble of any protein, trying to figure out how this disorder ensemble encode the function or regulation of this disorder proteins. But it's really expensive, and we really cannot use this atomic approach to study phase separation of disorder proteins. But if we could, train good generative model to become a surrogate MD simulation engine, this could be something extremely impactful. So I think that this is so if you guys are interested, this I think in my opinion is the most advanced, the most impactful work in this particular direction. So let me just go back to the.
Let me see if I can reproduce this correctly, because this is a tricky one.
It's really tricky one.
Generate a bunch of protein structures, train a LLM effectively, the generative model, on those structures, and then start hemorrhaging thousands or millions of maybe real, maybe hallucinating, but like plausible structures. And it's example, the space of possible structures. It's the space that this thing produces.
Yeah.
Okay. And any clue about the distributional properties, whether they're preserved?
So in terms of, I think at least the paper, the data is in the bioRxiv. You can probably just Google and you'll find it. So the limitation actually is not how well this pipeline will reproduce the ensemble distribution.
Mm-hmm.
The bottleneck actually is the quality of the input trajectory, because they have to use some simplified version of atomic model. So there's so much bias in the training data, and that actually is the key limitation of this. The ability of this AE/DDPM model to reproduce ensemble actually seems to be really decent. As far as I can tell, it generates ensembles essentially almost indistinguishable to the input trajectory.
Interesting. And it's important. It's not just pictures of the proteins. It's their little movies of their motion, right? Trajectories.
Yeah, it's trajectory, or it's ensemble. I mean, when I think about trajectory, I think about a time correlate movement, right? So these are not. These are really just snapshots, uncorrelated snapshots that you are generating from this pipeline.
Okay. And then if the bottleneck is generation of data from an atomic simulation, then of course you can use ML to speed up the atomic simulation so it can generate more, use up better.
You could think about pilot. So for me, right, so I feel that the biggest opportunity is to really how to bring in, right, transfer what we learned from predicting structured folded proteins, the structure of folded proteins into this pipeline. Right. I think it's where a lot of information can be leveraged to make a more a tool that can generate more realistic ensembles. Because right now this is really just trying to be a surrogate of MD engine, and then you're going to be limited by all the limitations that we have in our force field, in our MD simulation accuracy, so on and so forth.
Okay.
So this will get you a little faster, but I don't think you're going to get you very far.
Right. Makes sense. Thank you.
Yeah. So anyway, so I'll probably make this the last slide. So this CF model, right, as I try to convince you, is really, really simplistic, but this can be really, really useful. And we actually learn a lot of insights about how different sequence variation control phase separation, so on and so forth. It's really a fantastic model, but it's really, really overly simplified, right? So one thing I want to just emphasize is that if you think about protein as a polymer, the defining feature of the protein polymer is the backbone, the peptide plane. So which is really showing here. So peptide backbone have really unique way how they interact, and the propensity of the backbone really is a baseline determining the conformational property of the protein polymer. And this is really completely missed, completely obscured in this kind of CFR model. And the one important property I can highlight is that if you think about disorder protein, they are not random polymers. They have non-trivial local and long-range structures. And again, this cannot be described by the kind of CFR model. For example, people know experimentally, also computationally, people know that there are, for example, residual helices that can exist in the level, frequently exist in disorder ensemble. And the presence of these transient structures has important consequence in how this disorder chain will come together and interact in the condensed phase. And experimentally, it's also known that there will be, for example, this is something called the CD measurements, and so what it really detects is the backbone structure. Without getting into details, right? What it really shows here is that this particular protein called tau is a really important protein in many different things. Is that when they undergo phase separation, there is a clear change in the CD signal, indicating the secondary structure is different. So this is another work done by Nick Fuzzy's lab. They show that this other important protein, there are so many important proteins in biology. Anyway, they have this transient helices. They argue that this actually a really important role in mediating the interactions within the condensed phase. And this kind of detailed backbone interaction, the structures, how they model the phase separation is really something really, really challenging, but it's an important next step that we need to do in simulation of this type of approach. But we need to go beyond the CFR model. So I'll probably just stop here. And this essentially just going to say that when people trying to ask these more detailed questions, that at present the only choice is go all the way to atomic model. But this is extremely expensive. You can't really do anything. And in our lab, we have now worked on a new coarse-grained model we call hybrid resolution model, where we now have more atomic representation of the backbone, but the lower resolution, the side chain, such that we can parameterize this kind of model to provide a more realistic description of how backbone interacts and how they form transient structures and how the structural properties will contribute to phase separation. So I'll probably just stop here because there's so much detail that.
Of course, yeah. And actually, this is interesting because it sounds like now you're doing molecular dynamics, except instead of having traditional forces, these long-range directions that all fall off with a certain thing, rate, you have like crazy protein strings that touch each other, and it's a much more time-dependent floppy kind of force abstraction. Okay. And it would also be very data-driven because they're so much more complicated.
Yeah. So I would just point out that I think this is what I mentioned earlier on, respond to your question. Mm-hmm. So in design and the parameterization of these kind of models, oftentimes one of the most important limitations is that we do not have enough data to guide us in tuning all these detailed interactions. So it becomes really empirical. So oftentimes, for example, like some of for the disorder proteins, now they are actually, because it's such an important class of protein, now they are pretty high quality data on probably 50 or 60 sequence. Like when you nucleate as RNA, the data, the only data we can find actually is on 2 of them.
Okay.
The poly A and the poly U, and these detailed structural properties. These actually are really difficult data to have. So now it becomes really tricky on how you go about designing the coarse-grained scheme designing how you're going to optimize your parameter. It really need to be, you need to be really careful in confining yourself to the physical boundary, right? What's plausible. So it becomes really empirical and intuition driven.
Yeah.
So it can be a good thing, but it can be really dangerous, right? Because now it depends on what I believe, or depends on what you believe.
Right.
And by the end of the day, because of the limit data and limit resolution, it's really difficult to say, oh, my model is better than yours. You can't really tell per se.
Yeah. And a lot of modern ML is built in a lot of data, which means that to use it, you can't, these data sets are not good enough. You need to go from atomistic simulations is the only source of data you have, but they're slow.
Yeah. So I should also mention that I think one important impact, right, of this type of approach, like coarse-graining or the coarse-grained approach, like as I was trying to illustrate early on when I talk about clovers, is that so if you can tune the model that is somewhat reasonable, capture some of the essential physics, then what you enable to do is now you can really generate a lot of surrogate data. It's not fully realistic, it's not experiment data, but it contains some information or sufficient information. And those data can become really, really useful if you want to really build a more powerful machine learning model that can go back, because you can think about maybe there's a feedback loop, a pipeline you can think about doing in this kind of approach. And the one thing that I did not mention is that there's really a lot of interest in studying the material property of this condensate. So for one example is really hydrogel. So right now, if hydrogels are really important type of material people in wound healing, tissue engineering, and right now you have no control. Usually I come up with this peptide, you found a gel, it's like great, that's it. And if you say, okay, you make the gel more rigid, or can make gel less, nobody knows how to do with this. So, but if we are able to now have an approach, right, it can simulate, predict the properties, the molecular properties of the gel. Then you can start to think about, you can rational design, engineering, machine learning, all things can become plausible. So I think really there is a huge potential and the important need to really design and parameterize testing and using this different type of coarse-grained model in this domain.
Okay, that makes a lot of sense. Yeah, thank you very much. It was fascinating.
Yeah, thank you, and it's a lot of fun. Sorry, we did not get to go through any of the things that we work on, but I'm glad that at least I will introduce the problem, introduce the general approach, and the many of the possibilities.
Yeah, and actually, it's an excellent summary of the field. Yeah, thank you very much. Have a great rest of your day.
Yeah, great. Thank you, Greg.